Let's start off by having a look at the life cycle of a typical neurotransmitter molecule. Once released, a neurotransmitter molecule has one of 5 fates. Let's explore these fates step by step. First, and most importantly, a neurotransmitter molecule can travel across the synaptic cleft and bind to its dedicated receptor on the other side, the postsynaptic side. The synaptic cleft is a structure approximately 20 nanometers wide, which is about 5,000-fold smaller than the width of a human hair. On average, a neurotransmitter travels at a speed of 0.05 miles per hour, which is about 25 times faster than a common snail, but 200 times slower than the typical cyclist riding a bike. At this speed, it takes a neurotransmitter molecule less than 1 microsecond to traverse the synaptic cleft. That's 1/1,000,000th of a second. Pretty fast. By comparison, a synapse covers a very large surface area compared to its width. It would take a neurotransmitter molecule about 1 millisecond, or 1,000th of a second, to travel out of the synaptic cleft. This is clearly a significant difference, but you might ask yourself, so what? The answer is straightforward. Recall that the goal of synaptic transmission is not to only transmit information from A to B, but also to transmit that information exactly at and only at a specific time. This last point is the key. If a neurotransmitter were allowed to remain in the synaptic cleft for too long, it could bind and unbind to its dedicated receptor and potentially activate the postsynaptic cell multiple times before diffusing out of the synaptic cleft. As a consequence, the time-resolved transmission of information from neuron A to neuron B would be lost. Thus, mechanisms are needed to shut off neurotransmitter action in the synaptic cleft before simply diffusing away. 4 such mechanisms are known to date. First, after crossing the synaptic cleft and binding to its dedicated receptor, the neurotransmitter, and sometimes even the entire neurotransmitter and postsynaptic receptor protein complex, can be resorbed into the postsynaptic cell. Second, neurotransmitters can be broken down by specific enzymes that are present in the synaptic cleft. For example, the neurotransmitter acetylcholine, which you might recall from a previous lesson as the primary neurotransmitter acting in the neuromuscular junction, is cleaved by an enzyme called acetylcholinesterase directly in the synaptic cleft. Third, neurotransmitters can be taken up by specific transporter proteins present on the surface of glial cells that surround and even sometimes invade the synaptic cleft. This is a process of great current scientific interest. Finally, combinations of the aforementioned mechanisms are also possible. For example, acetylcholine can be cleaved by acetylcholinesterase directly in the synaptic cleft, followed by the uptake of one of the breakdown products, in this case choline, directly by the presynaptic nerve terminal. Much as we humans do with some of our trash, in the presynaptic neuron, these breakdown parts can be recycled to produce new acetylcholine neurotransmitter molecules and ready them for another life cycle.